In April 1998, the crystallographic structure of a bacterial potassium channel was unravelled by McKinnon and his colleagues. We present here some of the results of our theoretical calculations that make use of this new structural information. We show the protein structure of the potassium channel from outside of the cell, looking down at the pore region of the channel. The structure, determined by X-ray diffraction, consists of 396 amino acid residues, or approximately 6,000 atoms. The channel is constructed by 4 subunits of a tetramer of peptide chains, each represented here by different colours. Here we show just 2 of the subunits, each consisting of helical arrangements of amino acids represented by ribbons. Each subunit consists of an outer helix, inner helix, pore helix, and a selectivity filter. The protein atoms form a pore between these subunits. We follow the lining of this pore with the blue curve. We generate a cylindrically symmetric pore lining by rotating the 2 curves shown around the channel axis by 180 degrees. The channel is composed of 3 segments. A long, narrow segment of length 20 angstrom, lined with hydrophobic amino acids extending towards the intracellular space. A wide water-filled chamber of length 10 angstrom, and a narrow selectivity filter, approximately 12 angstrom in length and 2.8 angstrom in diameter, extending towards the extracellular space. The selectivity filter section of the conduit is lined with carbonyl oxygen atoms, shown here in red. A succession of carbonyl oxygen rings appears as an ion traverses the selectivity filter. The ion then traverses a region lined with mostly hydrophobic protein interacting with negatively charged amino acids near the channel mouth. Here, we illustrate the interactions experienced by a potassium ion as it enters the channel from the intracellular side of the membrane. First, the ion is attracted into the pore by negatively charged glutamic acids at the channel mouth. The field of the pore helix dipoles then pulls the ion into the wide chamber region. The ion then enters the narrow selectivity filter by discarding water molecules and interacting with the negatively charged carbonyl oxygen atoms lining the pore. Finally, the ion experiences the field of negatively charged aspartic acid residues as it leaves the channel and enters the extracellular space.